This session is again going to look at termination of eukaryotic DNA replication, but the focus will solely be on what is called as end replication problem, and that occurs at the ends of the linear DNA. What is reported and what we know today is that telomerases help in solving the end replication problem. So let us look at what are the learning outcomes of this session. Replication of the leading and the lagging strand being different. One of the main focus has been in ensuring that the Okazaki fragments are made continuous before termination begins. However, eukaryotic DNA being linear, the ends of the lagging strands are not completely replicated, leaving the double-stranded DNA with an overhang at its 3' end, and as a result of which one strand becomes shorter than the other. And therefore, with every replicating cycle, the DNA will become shorter, which can be detrimental. The ends of the chromosomes are found to have repeating sequences called telomeres, which are heterochromatin and can protect the DNA ends. So it is these telomeres that are extended by a special polymerase called telomerase, which eventually helps take care of the end replication problem. Now, let us first understand what do we really mean by end replication problem. So let's again go back to initiation of replication at the origins. So you can see that you have uh, replication forks being formed at every origin, and they begin to enable replication of DNA bidirectionally. And as the replication forks from two different origins move towards each other, they meet at what is called as a convergence zone, and you have termination occurring at the convergence zones, which we looked at in the last session. However, in this session, what we are going to focus on is mainly what happens at those ends of the linear DNA. How does termination happen at those ends, or is there something that is different from normal? So that is what we are going to look at in this session. Now, so what is end replication problem? So let us consider that you have a origin and therefore you have two replication forks, each moving in two different directions. Let us consider only just one part of the replication fork. So considering that this is the replication fork that is opening up the unreplicated DNA to get replicated DNA, and because of the DNA opening up, you have one strand being synthesized continuously and the other strand being synthesized in what is called as fragments, and those fragments are what are called as Okazaki fragments. So therefore, this strand that is being synthesized is what is called as the lagging strand synthesis. So you can see that as the fork moves forward, you will have the leading strand being synthesized continuously, while the lagging strand you can see has Okazaki fragments. And eventually, suppose you have the replication fork moving till the end of a linear DNA, what is going to happen? So what's going to happen is something like this, where you have the template that is used to form the leading strand complete, while in the other strand you have the Okazaki fragments, and these Okazaki fragments are being matured. So when you say matured, what do we mean? We mean that these primers have to be replaced, filled up, and then sealed. So considering that that has to be done, let us note that say for example the primers are removed. And on removing the primers, what therefore happens is, using this 3' OH, you would have the DNA polymerase filling up the gap. Same is considered over here. So you have these gaps where you had originally the RNA primers being filled up by the DNA polymerase. But at this end, the DNA polymerase does not find a 3' OH group. In both these cases, because DNA polymerase could use a 3' OH group which is present over there, it could extend the DNA and fill up the gaps. However, in this case where there was an RNA primer, now the DNA polymerase is unable to bind to anything to initiate replication. And therefore what happens is, at this point you are left with an overhang. So we know that 3' to 5' is being read, and so you have the synthesis of the lagging strand in the 5' to 3' direction. So therefore, in this newly synthesized DNA, you will not have this portion filled up, and so you can see that one strand of this replicated DNA is shorter than the other. So what you have over here is what is called as a 3' overhang. So with every replication cycle, so in the next replication cycle, these two will become the templates. So you can already see that one template is shorter than the other template. Now, when you have the lagging strand of these templates being synthesized, again you will have it getting shorter every cycle. So this is something that is a problem. This was actually identified by Watson as well as Olvinnikov in 1970s, and they had said that there is an issue with completing replication at the ends. And it was later on that they also realized, because Hayflick had mentioned long time when he was culturing cells in vitro, he found that cells could survive in culture only till certain divisions, after which what happened is the cells start senescing and dying. So therefore, that was a point of time where this was understood that cells cannot continuously remain, and it has a limit to which it can divide. So with every division, what possibly is happening is that the cell's ability to survive is decreasing. And therefore, that was something that was noted by Hayflick, and it was known as what is called as Hayflick's limit. So that there is an issue with the replication at the ends of the DNA, linear DNA, was understood. But what could be done of this end replication problem is the next thing. Now, what was found when people began looking at, what is present at the ends of the linear DNA, and one very interesting experiment also showed that the ends of the DNA were less subject to mutation when exposed to radiation. So that was something that created a lot of interest, and the scientists wanted to know what are the ends of the DNA made up of that enables it to protect itself from mutations. So when they studied these ends of the DNA in Tetrahymena as well as in yeast and other species, they found that most of the telomeres, you take any species, and in most of the telomeres, most of the DNA, chromosomal DNA, at the ends of the DNA, one would find what is called as telomeres. And these telomeres contained 20 to 70 tandem repeats of a sequence TTAGGG. So therefore, the sequence that is there is a GT-rich sequence, and therefore, when you have repeats of this GT-rich sequence happening, the entire region becomes what is called as the GT-rich regions. Now, these telomeres at their ends may have again an overhang present, and these overhangs can be anywhere between 30 to 300 nucleotide longs, and so those overhangs which are present at the telomeres are called as a G-tail. Why G-tail? Because they are rich in GT. Now, the actual repeated sequence and the number of repeats may of course vary from species to species. Say for example, this TTAGGG that you see is typically found in human telomeres. But when you go to look at the yeast telomeres, one finds that the sequence can be a little varied. Now, all eukaryotic chromosomes, therefore, at the ends, okay, they bend back on themselves to form what is called as a loop. So what you see over here is a human telomere or a human T loop, and the size of the T loop can vary depending on what is the length of the telomeres. Now, the size, as I mentioned, can vary. And the T loop formed is again stabilized further by a lot of multimeric proteins binding to it. And this forms what is called as the telomeric complex. Now let us just look to understand the telomeric region. Okay, what is the T-loop architecture? And the T-loop architecture has a complex that is called as Shelterin. The name Shelterin is because the entire complex helps in protecting the ends of the chromosomes. So when you look at the DNA, you will find that the telomeres begin from here. You can see that TTAGGG is present over here, and that is being repeated. Okay. And its complementary sequence is obviously going to be AATCCC. So in this strand, you have AATCCC, and in this strand, you would have TTAGGG. So any sequence that has complementarity to either this or this can invade this DNA. Just keep that in mind. Now what has been observed is that there are a lot of proteins that bind to this telomeric end, okay, and stabilize it to form what is called as a T-loop. Before that, do note that just as mentioned earlier, look at the end of the telomere. You will find that this part of the telomere is what is the 3' overhang, or what is called as a G tail. Okay. Now, to form the T loop, the G tail, which has TTAGGG, okay, because you know that the entire telomere is nothing but a repeat of the sequence. So the 3' end will also have the TTAGGG, and so therefore the 3' overhang is able to invade the telomeric sequences itself to form a hydrogen bonding with ATCCC and displace the existing strand. Now, therefore, what you observe temporarily over here, or what you observe when the 3' overhang or the 3' G tail binds to the telomeric region, you have what is called as a triple helical, a three DNA structure or a complex. So this is one DNA, this is the second DNA, this is the third strand. So temporarily here you will find that there are three DNA strands present in a complex. This being a single strand, because this 3' overhang has displaced the existing strand, this becomes single strand, and therefore this single strand is protected by what is called as protective protein. This is the protective telomere binding protein 1. Now, at various places, wherever there are the telomeric sequences, it has been found that there are two proteins, telomeric repeat factor binding protein TRF1 and TRF2, that binds to this double-stranded telomeric repeats. And they become the scaffold, or they recruit what is called as proteins that form the Shelterin complex. So you would have TRF1 and 2 recruit many other Shelterin proteins like TIN2. TIN2 is nothing but TRF interacting nucleoprotein 2. You will have TPP1, RAP1, etc. that all form what is called as the Shelterin complex. And together, what they do is that because they bind to the telomeric repeats, they are actually protecting the telomere. So they are basically forming a multimeric complex over here in such a way that the telomere itself with the 3' overhang loops on it. So here you do not have any longer any free end, and the free end which had the 3' end is now linked, while whatever displaced DNA is there is already also bound to by what is called as the POT1 protein. Now, therefore, this G tail is attached to the telomeric repeat. So, therefore, you can see that it is ensconced within the entire complex and is protected. And of course, you have the D loop form because it is invading the double-stranded DNA. Now we come to what is the function of the telomerase. So, since we know that you have a 3' overhang at the end of the linear DNA, And even at the telomeric end, you have the 3' overhang. It means the telomerase has just one job to do, is try to extend the telomere. And the longer the telomere region is, the more protected the genes, that is the unique functional genes, are. So considering that this is the scenario where you don't have a primer being filled up over here, and so you have an overhang formed over here, we will look at it a little in detail. So let's consider this is the template that is being read. Okay, so you'll find that the ends of the DNA always have the telomeric repeats. So you have TTA, GGG, TTA, GGG. This is a constant tandem repeat sequence. And on that, until you had the primer, so this is the region where you had the primer. That primer has been removed. Same, it may be removed by RNase H, but the DNA polymerase was able to synthesize the new strand only until here, and therefore you have this overhang present. So this is the 3' overhang. Now this portion is extended further by telomerase. So let us see how it does so. So the telomerase comes and binds to the 3' overhang that is there because of the end replication problem. And once the telomerase comes and binds, the most important thing that happens is that within the telomerase is present an intrinsic RNA sequence, and this intrinsic RNA sequence is complementary to the tandem repeats that are present. So it is complementary to the telomeric region. So this RNA that is present inside the telomerase plays a very important role in carrying out the extension of the telomeres. So please remember that the telomerase has a RNA within its structure. So you can call this as a ribonucleoprotein that acts as an enzyme. Now, having looked at this, therefore, you have all of this nothing but what is called as the telomere encoding RNA. So this part of the telomerase is called as TER. While the protein part, of which there are many domains, the catalytic domain is what is called as the telomerase reverse transcriptase. So therefore, the telomerase is a specialized polymerase which, instead of using DNA as a template, uses RNA as a template to synthesize DNA. And one of the major domains that is present is what is called as the RT domain. This RT domain, that is the reverse transcriptor domain, has a very typical palm and finger domain which is present in most DNA polymerases. The only difference being that this catalytic domain uses an RNA that is already existing within that as a template to synthesize DNA. So having considered that the telomerase comes and binds to the end of the telomeric region, so the overhang, basically you can see it is a repeat of CCC, AAU, CCC. So AAU, CCC is actually the telomeric sequence, complementary telomeric sequence. So before that you have what is called as a CCE that can bind to the GGG of the tandem repeat, and on binding to the GGG of the tandem repeat and this acting as a template, you would have the telomerase synthesizing the DNA. So you have what is formed as the TTA-GGG, and this becomes again an overhang for the telomerase to come and bind. So you can see earlier it was bound here, now it is bound over here, and now it is going to add again another repeat. So this way what you will find is that this 3' overhang that was present is getting more and more extended. Okay, and so you will come to a stage where you have a long stretch of the telomere that has been extended, and this is single stranded thanks to the telomerase using its own RNA as a template to synthesize it. Now once you have sufficient amount of the telomere formed, you would have a primase add an RNA primer, and using this RNA primer, a DNA polymerase using the 3' OH of that RNA primer will again synthesize the complementary DNA. So what happened is, where the DNA had ended over here, now you can see a further stretch of DNA synthesis thanks to the telomerase being able to extend the strand. So what you would definitely therefore have is again that this portion is no longer going to become shorter. In fact, it has now got extended further. So at this end again you will have an overhang which can be used as by the telomerase to continue to synthesize or to continue to extend the telomeric ends of the DNA. However, what has to be noted is that the telomere again has a limit to which it will extend it, and that is regulated. So there are several regulatory aspects of telomerase, the T-loop and the Shelterin complex that decides when the telomerase has to lengthen the telomeres. If one chromosomal DNA has been extended already, then the telomerase will not go and bind to that. Instead, it will go to a chromosomal DNA whose ends have not been extended. So that is something that is regulated. So let us conclude, therefore, at the ends of the linear DNA is formed a 3' overhang due to the inability of DNA polymerase to fill up the gap. This is what is the end replication problem. These ends are telomeres, which are tandem repeats, and these can be shortened with each cycle of replication, ultimately leading to genomic instability, not stability, instability. The telomerases solve this end replication problem by binding to the 3' overhang and extending it further. This extension enables primer synthesis and then synthesis by DNA polymerase as the RNA primer again provides the 3' OH. The telomerase is able to synthesize DNA as it has an inherent RNA, which it uses as a template, as it itself is a reverse transcriptase, which uses this RNA to synthesize the DNA. Thus, telomerase with its special reverse transcriptase are able to extend the telomeres, thereby stabilizing the chromosomes. The ends of the eukaryotic DNA are further protected by the formation of the T-loop and a complex around it, thereby also protecting the telomeric ends from various DNA damage agents. Thank you.